A very good morning to everyone. On behalf of Institute of Pharmacy, Sri Ram Sarup Memorial University, I, Avnish Verma, moderator of FDP, welcomes our today's speaker, Dr. Abhay Bharadwaj sir. Before starting the session, I would like to introduce Dr. Abhay Bharadwaj sir. Dr. Abhay Bharadwaj sir has completed his graduation BPharm degree in 2004 from Uttar Pradesh Technical University, Lucknow. He accomplished his post-graduation MPharm Pharmaceutical Chemistry from Uttar Pradesh Technical University, Lucknow in year 2006. He completed his PhD in Pharmaceutical Chemistry in 2017 from Vanस्थली Vidyapeeth, Rajasthan, Jaipur. In 2006, he worked as Daffodil Pharmaceuticals, Meerut as production officer. Then he joined the Department of Pharmaceutical Chemistry, NKBR College of Pharmacy, Meerut. In 2007 to 2010, he joined Translam Institute of Pharmaceutical Education and Research, Meerut, in capacity of senior lecturer. Presently, he is working at Kite Group of Institution as an associate professor. He is having 15 years of teaching experience of industry and academic. He published more than 22 works in national and international journals, 3 textbooks, and teaching ads and field 12 patents, and where 3 are published and 7 are granted. He has presented more than 35 works in various national and international seminars and conferences proceedings. Now I request to Dr. Abhay Bharadwaj sir, please start the session.
Thank you, Avnish ji, my good friend, for giving me a wonderful introduction. And first of all, I would like to thank Sri Ram Sarup Memorial University Institute of Pharmacy to give me this wonderful opportunity to be the part of this 5-day wonderful FDP program. And one more thing, one more member is there who is always behind us, our friend and we can say your director, Miss Dr. Manju. And I would like to thank also her for this wonderful opportunity for this session. So should I start the session? If everyone is there inside the room?
Yes, sir. Yes.
Okay. My slides are shared with you.
Yes, sir.
So the topic is a very known topic, that is in-silico drug design. But we can say it is not least topic, because each and every day the things are going mature over here. Newer molecule is coming day by day. And again, the problem is that why we are going to be turned up on in-silico drug design. So one by one, we will cover each and everything. Wherever you have any query, you can write. Then I'll explain in the last. So we have some outlines of the session, like we will approach drug, then why in-silico, what are the role of in-silico in drug discovery and development, what are the various approaches, where we will explain about the traditional as well as the newer approaches. Then one special session is there, which is a requirement of time, that is in-silico hands-on training. Here we will discuss some online software by which we can predict some parameter of a newer drug molecule, or we can say the unknown drug molecule. then finalize with the docking, and we will discuss some success story over here. So we know very well, being a pharmacist, being a pharma professional, a drug molecule is a very important key ingredient with we people. If the person will take food, definitely the another thing is a drug. If he and she is not in any kind of disorder or disease, so the drug is very important, and we can get it from the different source. The source could be animal, plant, inorganic, synthetic. But right now we are more dependent on the synthetic molecules, because already we have we can say churned the number of molecules from the side of animal, plant, and inorganic sources. So right now the current era dependency is more on the synthetic processes. So here we are developing or discovering newer molecule in respect of the drug design. So as we are just thinking about the drug, so drug is a particular thing which will be respond on some receivers, and the approach of drug could be in the same respect. Like if we are just making some newer molecule or any we can say the pharmacophore, so definitely we can think about which kind of receiver we are taking. It could be enzyme, receptor, it could be any antibody. So there are number of receivers. In respect of these, we can prepare a drug. So we can say we have the ample opportunity to make the drug. But again, that journey is not easy as we are predicting. Why it is not easy? Basically, in this current era, the drugs are very much needed. Why it is needed? Because we have some cases, special cases, where the medical needs are more to be meet with them. Like we have some new diseases, as we know very well, the last 2 years that was generally covered with the corona virus. Swine flu is there, AIDS, we are not getting the good results over there. We don't have any kind of good promising molecule over there which can work up to this aid. Then we have some low efficacy of drugs, like in dementia cancers. Again, the cancer is a life-threatening disease. Side effects, already we are just fighting with the side effects of drug. And on the sake of the better responses, we are generally undergoing the side effects. So again, these all things, like somewhere the cost effectiveness is there, like the spinal injury requires the more cost in the treatment. Likewise, any kind of hormonal therapy is, again, it's very costly. So again, there is a requirement, some replacement. Drug resistance, major causes there. Antibiotics are generally suffering from this particular problem. Sustain industry activities. The newer molecule is also required in the generation, because most of the persons are generally taking their bread and butter with the help of industry. So we can say a large number of populations are working over there for their survival. That's why we need a newer molecule every time. Okay, these are the basic approaches. These are the basic needs where we have to work for a new molecule. So again, we have the two questions. The question number one, what is in-silico drug design? Why we are going towards? Another one, what is the need? What is the actually need of this science? So from this point of view, why? What is the need? What is the requirement? What is the necessity of this in-silico drug design? So basically, for this particular explanation, or we can say description, we have to take some backlogs, and which are just like that. If we will just keep a keen look on a drug discovery and development, so here there is a very long journey, which kind, like in that particular process where we are explaining a molecule is promising in any regard, in any kind of activity, it takes a long time. It takes a long time, and very well we will be dependent on the traditional technique where we are synthesizing a single molecule, and that molecule is not at that par. It will be active or inactive at the last. Okay, likewise, we are expending more than 800 millions. My dear colleagues, a single million have around 10 lakh rupees. Here, the story of 800 million, and nowadays it is going improve, as we know very well. The GST is going increase day by day. The cost effectiveness is going increase. Investment is going increase. So that value is also increased over here. So again, we can say that particular journey requires more time and more investment. That's why if we are synthesizing a single molecule in the same time, it could be not beneficial for we people. So definitely for overcoming such type of problem, we have to shift some hyphenated techniques, some advanced technique, and we can fulfill the requirement of the particular population from the point of view of newer drug material. As we know very well, we have some traditional approach where we are working last from 2, 3, and 4 decades. Likewise, serendipitously, a drug. Already we have the set good examples of serendipity, means any kind of invention which are just going to by chance or by accident. Already we have the some good example of that. Random screening, chemical modification, and the right now we are shifting on the rational drug design, or we can say the rational drug design approach. That is a newer approach. So primarily we will discuss a bit about the traditional approaches, then after I'll come on the newer approach. So as I said, and any kind of invention by the accident is generally known as serendipity. And we have, as you can see in the pictorial presentation, there is a doctor. He just masks their eyes with the cover, and he is selecting some material, some particular medicine or ingredient, and he is mixing something inside the container. So it is kind of serendipity where we are mixing 2 and 3 things, and suddenly if we are getting any kind of active promising molecule. So that was the history of we can say serendipity, where we got the drugs like we have the drug propanolol, and serendipitously the proctanolol drug molecule are generally comes through. Likewise, the sulfonamides are come from the prontosil dye. That was a serendipity at that time. We have the nitrogen mustards as an active drug molecule in respect of cancer. Random screening, the another part of traditional, we can say, the drug design, that is a random screening. In this particular zone, the screening molecules are generally large, where we are selecting 2 and 3. And finally, we will decide the molecule as per their screening. So again, these all approaches that was time consuming. It requires the more trials. We can say the hard work is required over there. The cost were more effective over there. So we can say right now these approaches are going to deplete, and we are going to be shift on the new technique, because each and every day the time is running very fast. The newer ailment and disease are coming inside the market. Each and every disease and disorders require the newer molecule. So again, to meet this requirement, we have to shift on the different approach. So now, what is the In-Silico design? So as you can see into the, we can say in the slide, the In-Silico means the experimentation performed by the computer. Means each and everything which is related to the drug design, where we are synthesizing, where we are preparing, where we are modifying, where we are just inculcating anything inside the molecule, basic molecule, pharmacophore, that all the process, all the work are generally computer dependent. And we know very well, if the computer is there, definitely the softwares are working with them. So we have the different approaches with the help of different software, and we are utilizing this approach. Why in silico drug design is needed? Again, the second question is coming. Why in silico drug design is needed? So that was a simple, we can say, you can see the stairs which are going to be start from the thousand of molecule, where the different parameters are underlying, like target, selectivity, potentiability, permeability of drug molecule. So the number of parameter are working with them. Some will be fail on the target, some will be fail on the selectivity, some will be fail on the durability, stability, non-induction. The particular sex differences are there where it will not work. Some, we can say, some are not capable for the pregnant movements. Some will not cross the placenta, some will not cross the blood-brain barrier. So there are the different, we can say, parameter or approach where we have to, we can say, potentiate our drug molecule. Then what we are seeing over here, only 1 and 2 molecule could be active or promising at the end of this journey. So again, if we are identifying the number of parameters, then it is not going to be active, then the overall workup, overall labor will be zero over here in this step. So that step was very much, we can say, crucial for the medicinal chemist or the particular, we can say, the pharmaceutical worker which are working on the drug design or drug discovery. So again, to overcoming this problem, we are shifted on the computational approach. As we know very well, in the traditional screening, if we are going for the clinical trial, phase 1 trial, phase 2 trials, so it requires some animal, it requires some, we can say, the volunteer models, it requires some other commodity for the detection. So each and everything have their own cost, and just because of this cost problem, again we can say the drug permeability or drug approach is going, we can say, decrease day by day. So basically what's happened over here, if we are targeting some selective molecule in this particular in silico approach, so the molecule could be turned up into the library of compound. So there are some various sub branches which are working with the in silico genes, and we are utilizing these all genes on the computer software. So here we have the number of targets we can approach in any directions, like in life-threatening disease, AIDS, coronavirus is there right now. The pandemic is not going to be endemic. The research is going on, molecules are coming inside the market, vaccines are coming inside the market. So each and every day we are working with the newer techniques, newer approaches, with the help of these computational techniques. Again, if we are thinking, we are utilizing the computational approach, likewise, the CAD is mentioned over here. This CAD will clear about the many things. If we have the two type of molecule right now. One could be a known compound, another one could be the unknown compound. So the things are very clear about the known molecule. The approach will be, we can say, very simple with them. We can modify the molecule, we can add something, we can detect something, and we can finalize the newer pharmacophore. But the problem is that if any kind of newer molecule is going to be approached or going to be introduced, so how we can predict their activity? And activity is a very simple word to say, but it have the very huge gravity. Likewise, if we are thinking about the molecule, the first parameter, or we can say the very much important parameter, is that already I have seen you a slide where I am mentioning, in any discovery or any invention of any newer molecule, it requires around 800 million of the, we can say, investment will be there. But if we will think about, if any drug or any newer molecule is going to be synthesized, around 3/4 of the total cost are generally going towards the safety measures. safety measures, we can say a molecule, we can, the particular approach of drug design is like that our molecule should be effective or not effective, but it should not be toxic. It should not have any kind of adverse effect. It should not have any kind of side effect. So basically the maximum investment or maximum money consumption are generally based towards the side of safety measures. A drug should be safe for each and every person. We have some compromising target. Likewise, if the person is suffering from the cancer, so definitely some we can say some healthy cells could be compromised over there, but it's a need of treatment. But once we are synthesizing or approaching for a newer molecule, so it is mandatory, it is very much, we can say, necessary, the molecule should be devoid of such type of problems. So for this approach, this particular in silico drug design or the computational drug design is very much helpful for we people. It is giving us the robustness as well as the reproducibility. Means a same molecule, if we are utilizing, it will provide us the same results. There will be no any kind of deflection will be there. Library of compound could be used in the same approach, in the same time. We can predict, we can identify, we can optimize, we can screen a number of molecule library of compound. But if we are just think about the ancient era, a single molecule could be screened out in a single time. That was the problem of traditional methods. Fast prediction for a large set of compound in a same time, in a single time, or we can say in a very less of time, we can predict the various parameter of a known molecule as well as known molecule over here. Number of parameter we can detect simultaneously. Means if we are going for the pharmacokinetic parameters, so we know very well about the log p value is there, then drug likeness is there, then robustness is there, and physical parameters is there, drug solubility is there, steric parameters is there. So these all parameters simultaneously we can detect once we are going to just utilizing any skeleton, any pharmacophore of the molecule. We can predict the novel therapeutic targets also. As we are working on the software, so there is a freeness over there where we can generate something new. Likewise, we can predict the newer structure of new receptor. Already you know very well, we have the number of receptor inside the body. Some are enzymatic, some are nucleoprotein receptor, G coupled protein receptors are there, but there are so many receptor where we are not gone through. So again, with the help of this approach, we can identify those receptor also. Then that method is really, it's an economic method. Primarily we have to approach the software, then after there is no cost. And if we are just saying about the online connection, so we can predict some parameter with the help of online parameters where we have to pay off net expenditure only. So the modern drug discovery where we are utilizing this in silico approach, it is working like that, where we are identifying the target simply, target validation, lead identification, lead optimization, preclinical phase, drug discovery. So here the journey is going short with the help of this computational technique, or we can say the more promising journey is going on. Here the hits, have you heard about the hit? HIT. Hit means that is an active molecule. Okay, so active molecule, if we have the active molecule, so definitely we have some idea, we have some, we can say the chances it could be a positive molecule at the time of drug discovery. So again, we have the library of molecule or the number of molecule, the number is going increase. That's why we are more, we can say, positive. Our journey or our particular research could be positive at the end of this particular drug discovery session. So it is more beneficial in comparison to last one. So already some other approaches are combined with this like virtual screening we have with the help of docking, high throughput screening where we can screen more than 1 lakh compounds simultaneously. We can say randomly we can take some molecule from genomic, proteomics or bioform. We can apply the combinatorial chemistry where we can synthesize the library of compound with the help of solid phase synthesis, liquid phase synthesis. Already we know very well. Then molecular modeling with the help of computational graphics software, we can modify the molecule as per our requirement. Please unmute your mic. Please mute your mics, everyone.
आरएम का जब देंगे तब बना लेंगे, आरएम का तो अभी जब दिया नहीं है तब फिर क्या?
Pooja ma'am, your voice is coming. Please mute your mic. Okay. So here we have some simple parameter by which we will do some exercise, which are coming inside the In-Silico hands-on training. So with the help of this, we can say software technique or computer-based technique, we can identify primarily we have some parameter by which we can say this molecule could be active, could be promising, could be beneficial. It could be work against the any disease or disorder. So it is a prelim exercise which is very necessary in any molecule. Okay, as I said, we have two type of molecule: known molecule where we have some idea, and if we are altering or modifying the known molecule, again it will be categorized into the unknown molecule because already we have modified that. Okay, and the another section is the unknown molecule. So unknown molecule where we do not know about anything, the overall things is in blind, or we can say the gray area is there. So again there is more requirement to take a chance because there is more chances of success over there. So these are the parameters where we can work immediately with the help of our softwares like ADME profile, physicochemical parameter of a drug molecule, pharmacological activity prediction, it could be active, it could be toxicology, reaction, and finally drug receptor binding probability. Okay, so here I have mentioned the 6 parameter, and if we will apply these all parameter in regards of any unknown molecule, so we can say that molecule or the results of the molecule will clear about us it should be go for the next phase or not. So these parameter can clear us that molecule could be promising or not. So we can say it's essential parameters. That's why we are working primarily or preliminary. So now I am everyone is listening me. Yes, my voice is audible or not? Please away me.
Yes, sir, audible.
Okay, thank you. And that is an important parameter, which is, we can say it will help you in any kind of prediction. I know very well the faculty members is there, and these all faculty members from some person from the side of pharmacognosy, pharmacology, as well as pharmaceutical chemistry and pharmaceutics. You all are going through the research each and every day. You are just taking some challenges in your life. So these all sites, these all approaches will be beneficial in the prediction of any kind of activity. Okay, whether we are synthesizing, formulating, we are extracting anything, or we are just going for the we can say final screening of molecule. In all the branches, these all parameters are necessary. So I have some we can say way by which I'll clear you how we can got these parameters with the help of online software approach. So the software will be working simultaneously and it will finalize the number of parameter of drug. Okay. So if the screen is shared to everyone, you can see the number of parameter of the molecule. The first first molecule is paracetamol, which is showing over here. Then another molecule is naphthalene, which we have pasted over there as their smileys. And all the parameters are going to be open in the regard of paracetamol and naphthalene. So you can get the log S value, solubility, water solubility, pharmacokinetic parameters. The number of parameter will be open as per the Lipinski rule. The drug likeness is here, Ghosh rule, Weber rule. So there are so many things: lipophilicity of drug molecule, physicochemical parameter of drug molecule, all the physicochemical parameter in regards of ADME profile, which will open with the help of this software. Are you getting or not?
Yes, sir.
Okay. So this parameter you can simply retrieve from here. You can say we can just see over here the retrieve data, and two type of file is coming here. One is Excel, another one is CVS file. So both files will provide data from this side. You can copy your data with the help of the CVS. Once you will click on the CVS, so all the data will be copied over here and it will be directly shipped in your CVS file, which will be like a Excel file. Okay. Any doubt regarding, any question regarding this? Yes. Any question?
सर वन थिंग आई वांट टू आस्क कि क्या हमको ये मतलब पहले सॉफ्टवेयर परचेज करना पड़ेगा?
नहीं sir, this software is free for we all. Okay. Only we have to mention that particular site, that is swissadme.cs.index.php. Okay. Once you will simply add this site inside the address bar, the simultaneously site will be open. You have to paste smiley over here, then you can do your work. No problem. So that was the first parameter. Now I am coming on the next one. Screen is shared with everyone or not? You all are able to see the slide? Slides
right now,
नजर आ रही हैं आपको?
No slide is opening.
Just a minute.
Not showing slide.
Okay, just a minute, sir. वो actually हर उसमें ना shift करना पड़ता है, इसमें थोड़ा सा it's a problem of Zoom. No problem. I am just sharing again. Now it is visible. The slides are visible.
Yes, sir. Now
visible.
The person who is doing PhD, so some parameter you can do with these all we can say online citations. There is no problem. Now, if you are going to predict any kind of activity of a molecule, okay, if you are synthesizing, if you are extracting out any newer molecule, so if you want to predict any activity of the molecule, you can approach this another we can say the online software approach, way to drug. So the another software will open in front of you, that is way to drug. आप लोगों को दिखाई दे रहा होगा? If I am not.
yes, it is visible.
Okay.
Sir.
It is visible. So it is a particular software which will educate you about the any kind of drug activity prediction. Means this molecule, this structure could be active or not. So before start, you can see there is an icon, go for prediction. We have to simply click over here and it will ask about the login ID. So once you will start this software, you can register yourself and there is no more problem. You can easily register with the help of your email ID, as well as you can mention your password, your known password. Then you can undergo inside the overall software. So again, the pass online will help you to predict the activity. Primarily, you have to click on this icon, predict new compound. Simply you have to click. Then the further icon will be open like that, smileys, MOL file, Mervin files. So as we know very well, primarily we have designed the smileys of molecule. So we have to click on a smiley. The simple smiley is one of the best way by which we can approach 4 to 5 parameter of any newer or unknown molecule. Okay, the problem is there. If you have the known compound, you can take the smileys from PubChem also. Excite a PubChem, directly you can approach the smileys from there. Only you have to mention the name of drug like paracetamol, metronidazole, tinidazole, whatever. Whenever you will mention over there, you can get the smiley. But if you have any unknown structure, so again there is a problem. That's why you have to draw the structure on CamDraw and CamSketch, and you have to go likewise as I go on the tools, then generation, then finally smile. So again you have to click on a smile, then you have to insert or copy your paste your smile over here, then finalize as get prediction. Just a sec. So I have pasted, then you can see the newer table is going to be open. That is a smiley of paracetamol, whatever I have mentioned inside the given, we can say format, and we can predict the activity. So dear members of this FDP, you can see we have the two criteria which are shown over here on the screen. One is the PA and another one is PI. The PA reflects the activity of molecule and PI reflects the inhibition of the molecule. So here we have mentioned the known molecule. If you will mention any unknown molecule or any, we can say, synthesized molecule, then again you can predict about the activity or inhibition of molecule. So in the same front, you can see the number of things are coming, like the membrane integrity agonist activity, cytochrome P450 substrate activity. So here the activity will show like that. If the score is coming in the 9, means the activity is good. If the score is coming in a form of like that minus sense, then again the inhibition is more over here. So that molecule or that paracetamol molecule is active in that particular area, and it is showing their inhibitness or inhibity in this membrane integrity agonist with this particular digital representation. So which activity will be higher, which activity will be more promising, more potential, it will be comes on a top, and which will be least active activity, it will be comes on the, we can say, bottom side. One more thing, these all are the nodes, these all are the receptor, these all are the genes. Sometime it could be enzyme, sometime it could be nucleic protein, sometime it could be a receptor which are present on the human body or any kind of animal body, and that thing are generally reflect in a form of activity as well as inhibition of drug molecule. So that particular we can say online software pass online will clear about the activity of drug molecule. Now I'm coming back on the another prediction. Now the third approach is in regards of toxicity. So the third parameter is toxicity, and for this particular approach we have to go through the new we can say software, online software, that is Protox a second, which are showing here. I'm simply clicking over here as per the hyperlink. So I think the third software, online software, it is going to be open in front of you. ऑनलाइन सॉफ्टवेयर इज ओपनिंग, खुल रहा है आपके सामने. एनीवन प्लीज आंसर.
अभी सर ओपन नहीं हुआ.
ओके, जस्ट अ मिनट. मेरे हिसाब से तो होना चाहिए. इट इज गोइंग ओपन टुवर्ड्स माय साइड, नॉट शोइंग टुवर्ड्स यू. Now it is showing. Now it is showing.
Yes sir.
Yes, sir, it is visible now.
So you can see already I have opened the third software, online software. It will generally meant for the toxicity prediction. We can predict the toxicity of any kind of unknown molecule with the help of this. The simple icon is coming on the front, that is tox prediction. We have to click on that. Then you can see the two particular further sub icon is coming over here. The first one is PubChem name, another one is smileys. Okay, if you have the name of drug, then again you can mention the name, paracetamol. Okay, you can mention the paracetamol and you can go for search. Okay, so once you will go click the button of search, you can see the paracetamol structure is coming over here. There is no problem. And again, if you want to go through the smileys, then again you have to paste your smileys over here. Then again you can go for the smiley search. So with the help of smiley, the same molecular structure will be formed in the second this blog. Sorry. This blog, and once the structure will be reflect over here, then you can go which kind of toxicity you have to check with the help of this software. So my dear colleagues, you can see there are number of toxicity which are showing over here. Organ toxicity of endpoints like carcinogenicity, immunotoxicity, mutagenicity, cytotoxicity is there. There are some nuclear receptor signal pathway related toxicity. So there are number of toxicity which we can approach simultaneously, or we can say in a same time. So I am clicking on the first 5. Okay, then you can see the another icon where the start talk prediction words are mentioned over here. We have to simply click over here. Then the another site will open which will clear about the toxicity parameter of the drug molecule. So already we have clicked on the 5 positively. So the organ toxicity parameter is active over here. Okay. In last 2 slides, you can retrieve data by simple copy and paste. Already I have clear about you the ADME as well as the activity prediction. You can simply copy the data and you can paste in the Excel for your we can say observation or result. So you can see the organ toxicity is active over here, but once we will see the hepatotoxicity, paracetamol have the hepatotoxic action. That's why in a longer time it's a hepatoprotective, but carcinogenicity is inactive, immunotoxicity is inactive, mutagenicity is inactive, then cytotoxicity is inactive. So on behalf of this prediction, your molecule could be moved further. It's a simple parameter, simple way by which you can get the toxicity, any kind of toxicity parameter of your drug molecule. Okay, any question up to this? Any question? Okay, no problem. So that is a very simple way to predict the things, or we can say the toxicity of a molecule. When two things are coming here, average similarity 100% and prediction accuracy 100%. These two parameters are also clear us about that molecule is also the part of existing molecule. Means that molecule is in the existence, it is in the market. That's why it is showing the 100% average similarity as well as the accuracy. Now again back on the slides. The last site is, or we can say the last parameter is the adverse reaction parameter. Again, I am just clicking on the online software.
Sir, sorry for interruption. 1 minute
Yes.
is remaining.
So 1
After
minute.
that, we should start new session.
So I'll join, no problem, sir. I will join again. So the new online software is going open in front of you, as you can see. I think it is showing to everyone. देख पा रहे हैं आप लोग?
Yes, sir.
हाँ sir.
Okay. Okay. The software are bit similar. First one was way to drug. That was for the activity prediction. But it is a second one, that is way to drug adverse prep. That is for the adverse reaction. So again, you can see the smileys options are coming. We have to simply paste the smileys over here. Same. Then again, we have to click on the make prediction. So the software will work and it will clear us about. So some part was. So you can see in this online software for adverse reaction, we have simply pasted the smile of paracetamol and it provides us the specific parameter in regards of activity as well as inhibition of side effects like nephropathicity, then myocardial infarction and cardiac failure. So which kind of adverse reaction will be happen with the molecule? It simply reflect over here, and from this save CVS, save PDF way you can save your data. So that was the last approach from the side of online, and by which you can get any kind of adverse reaction of any molecule. Any molecule means any new molecule as well as any unknown molecule which you will synthesize in the lab. Any question up to this? Okay, and the last parameter, it is not a simple word. It is a drug receptor binding probability. Okay, once we will go through from the all four parameters, we can just take an idea virtually with the help of computer without expensing any single penny. We can, no money required for this. We are just predicting the things or approaching the parameter with the help of online software or the computational approaches. But the last parameter where we have to signify is if the drug is going to be introduced or it is going inside the body, then what will be the receptor response over there. So that is generally done by the docking. It's a simple, we can say the software, number of softwares are available over here by which we can get the score, docking score, and we can predict about how the molecule will be, we can say, beneficial for the receptor or not. Means we have the two type of resultant. First one is the association drug receptor complex, ligand drug complex. Another one is the dissociation of ligand and receptor, or drug and receptor. So both things are very much necessary. The drug should be bind and it should be dissociate also. Then when it will be dissociate, that time really matters for we people. When if the time of binding will be increased, or the particular interaction will be there for a more time, then the drug could be approached towards the side of adverse effect as well as the toxicity. So we can say it is very important parameter, vital parameter for B people to be considered as a active molecule or the promising molecule. So that is a computational approach by which we can predict the kilocalorie energies prediction of a drug molecule in between the drug receptor interaction as well as bonding. Okay, so it's an experimental method. We can approach with the help of different way, like we can approach the receptor, we can approach the ligand. Already you know very well about structure based or receptor based drug designings are there. Then we can dock the protein docking. Then we can dock the protein ligand. Protein docking means if we are utilizing any protein as a drug molecule, then after we'll go for the protein dock. Then if we are utilizing any chemical entity as a drug molecule or ligand, then we'll go for the protein ligand dock. Then what are the docking and scoring? Scoring is a result by which we can get, there is some minimum requirement, or we can say the standard value of the drug receptor interaction in a form of kilocalorie per mole energy. It is a kind of energy in between the drug receptor, we can say bonding or association, which will clear us about for how much time or for a long it will be associated with the receptor. So it is very much necessary to be predict, to be identified, to be evaluated in a favor of the drug efficiency or efficacy. Okay, so we will also go through the scoring for finalizing the best drug receptor model. Okay, so it is also known as best pose in between the drug and receptor interaction. So these are the various receptors which are generally working in a current era for the docking of ligand and receptors. We have some applications by which we can say we are generating the newer molecule, like we can generally predict or we can finalize the lowest energy. The lowest energy are generally in between the interactions. It is not a bonding. There is the two different word. The bonds which will be for the long, like covalent bond, coordination bond. These bonds are generally for the long time. And if the bond will be there for a long time, it could be produce the suicidal bonding. So we do not need over here. We need a lowest energy interaction by which a drug will be associated early and it will leave the receptor also early. So it is an approach of the docking. Then we can study the geometry of the overall complex with the help of this docking procedure. Library of compound we can design with the help of this docking methods, because basically that approach will work vice versa, whether in respect of ligand or in respect of our receptor also. So we have the good applications. Less time expensing, we can apply the high throughput screening. Fast and conventional screening, we can get the we can say we can scan the library of molecule within a time. So again, we can say it's a very beneficial. We have some success story also, like dorzolamide. It is a diuretic in HIV case, we have scanner that is generally go to this docking procedure. Then finally conclusion, if we are applying this approach, then we can get the good molecules, we can utilize the less time. The drug, as we know very well, once we are just going to predict a new molecule, we are just going to design a new molecule. So the 2-3 approach are generally there. The drug should be minimum in amount, it should be less expensive, and it should be effective for the we can say age group, or we can say the number of races, number of population could be utilized the drug molecule. So all this thing we can finalize with the help of this in-silico technique, because it is taking less time and it is providing a, we can say, a mammoth pool of drug to we people. So that is whole about the presentation. Now it's a time of question. So you all are free to ask the question which is related to my topic. Thank you, everyone.
Okay, thank you, sir, for your so much wonderful session. Sir explained very well modern aspect of new drug discovery. If anybody want to ask any question, please ask. Participants, you can ask question.
Even some chemistry student can ask some questions. Chemistry faculty members can ask some questions. There is no problem.
हेलो
Yes, yes.
सर, एक्चुअली मुझे एक चीज जाननी है कि इसमें एक कॉम्फा एक कॉम्सिया होता है सर, तो
जी,
उसका एक्चुअल में सर रोल क्या होते हैं इसमें न्यू ड्रग डिस्कवरी में? कॉम्फा एंड कॉम्सिया. हेलो
हेलो
सर, आपको मेरी वॉइस आ रही है?
Okay, thank you, sir, for such a wonderful session.